It's really nice to be sort of with you at least virtually. Yeah, so I'm Liz Stuart. I'm at Johns Hopkins. I was saying earlier, I'm currently in the Department of Mental Health with a joint appointment in biostat and moving over to biostat in July. And the work I'll talk about today is really kind of the type of work I do a lot, which is really bridging kind of methods and application to mental health. And so there's not going to be a lot of formulas and, kind of not going to be a lot of like super deep statistics, but I hope it's interesting from sort of how do we really think about the use of statistics to answer applied questions in mental health and sort of deal with the challenges that come up in real data analysis. It's also, I'm a little sad not to be there in person. I spent my junior year at Oxford and loved living in that general region and spent a lot of time in London. So anyway, very jealous of you all, but maybe someday I'll make it over in person. Okay, I want to very much first, and just to set the stage, I will aim to finish up like 45 past the hour or so to leave time for questions. I do have the chat open, and if there's sort of a quick question along the way, feel free to raise your hand or put it in the chat, but I'll leave time at the end as well. So just want to acknowledge really great support from PhD students, in particular, Carly Lupton-Brantner has done much of the work that I'm going to be talking about today. And then Leon DeStefano is another student working on sort of some Bayesian versions of some of what I'm talking about. Collaborators at Duke and here at Hopkins, and funding from the National Institute of Mental Health and PCORI, which is another sort of government funding agency here in the US. And I'll talk more about the data, but some clinical trial data from through a source called Vivli. Okay, so most of my work is on causal inference, so estimating the effects of programs and policies and interventions. And in that field, and much of my work sort of to this point has been either sort of non-experimental studies and things like propensity score methods, as well as some work on randomized trials and generalizing results from trials to target populations. This work is sort of bridging those and bringing in this idea of kind of predicting treatment effects for, not so much for individual, sort of for individual people, but understanding treatment effect heterogeneity. And for example, the National Institute of Mental Health, whenever they fund a randomized trial of a mental health treatment, they require people to include examination of mediation and moderation. So what I'm talking about today is what they call moderation. Again, this idea of treatment effect heterogeneity, or sometimes called modification, moderation. But fundamentally, it's about, do some treatment causal effects vary across individuals? And kind of ideally, people would love to use that to inform treatment decisions for individuals. So I call this sort of the holy grail. The problem is that this is really challenging in practice. So we typically think of randomized trials as the gold standard for estimating causal effects. They have the benefit of the randomization gives us formal unbiasedness of treatment effect estimates. We can look at subgroup effects in trials. We can see, is this more or less effective for people, say, with different co-occurring conditions or different levels of severity. But a major problem in randomized trials is that they are really always only statistically powered to detect an overall main effect. People usually enroll just the number of people to estimate sort of an overall average effect and have power to estimate that well. There's sort of this very rough rule of thumb that to be able to see whether there's effect heterogeneity, even just across two groups, say old and young, you have to have 4 times the sample size as you do for the overall main effect. So trials are great, but probably not going to be the only solution to this treatment effect heterogeneity question. So then we might think about, well, can we bring in non-experimental studies? And by that, I mean using, say, large scale, maybe electronic health records, which I'll talk a little bit about today, or other sort of large scale data sources where they might have a large number of people. They might reflect sort of real world use in terms of like practice in, actual clinical practice rather than in a trial context. They might have more representative population, they're sort of more reflecting that regular use. But of course, the problem is that they likely suffer from confounding, where the people who take the treatment or not are likely different from one another. And I see the note in the chat about, yes, I agree, we can't actually estimate individual effects. So here, well, I think it's like in a couple slides, I will formalize what we mean. But yeah, I would actually even argue that crossover experiments don't really directly get at an individual effect, because you also have the change over time. So we basically can never observe both potential outcomes for a given person at a given point in time. But we're going to be thinking here more about sort of average effects conditional on a set of covariates. So I'll formalize that in a couple slides. Yes, and regression to the mean exactly is one of the problems of why we have to be careful to not attribute a change over time as a causal effect. So the goal of this work here is, can we get the best of both worlds? Sort of how can we, although it might be the worst of both worlds too. And again, sort of let me set out by saying that really this work is motivated because many, many fields are trying to understand this treatment effect heterogeneity. And I think as statisticians, and sort of myself as a statistician, a lot of my interest is in sort of how do we help people figure out the best ways to do what they want to do, but also recognize the challenges in doing that and sort of be realistic about what we can and can't learn. And I'll come back to that later. So basically, fundamentally, the question is, can we combine the unbiasedness that we have in trials with the large size and representativeness of non-experimental studies? I want to note that this is a hugely growing research area. It's one of those fields where I feel like every 2 weeks I get, table of contents in my inbox that has new papers on this general topic. And there's people doing like different variations on it, trials, non-experimental data, et cetera. It also goes by a huge number of names, this sort of idea of combining data sources, so data fusion, data integration, hybrid designs, individual patient data meta-analysis. So and I'm not in this talk trying to summarize all of that. I'll cite later a nice review that Carly did. But anyway, it's a really rapidly growing area in statistics. What I'm going to be talking about today is a particular angle on this, which is this point of trying to combine multiple trials to better estimate treatment effect or better examine treatment effect heterogeneity. I will also sort of signal how to bring in the non-experimental data, in particular some electronic health record data, but also will signal again sort of some of the real world challenges that come up when we're trying to do that. Okay, so let me make it a little bit more concrete by giving you a little bit of background on the motivating context. So the motivating applied context is whether medications for depression are sort of differentially effective. Again, whether these medications might help some individuals more than others. In particular, we're going to be comparing two medications for individuals with major depressive disorder. One is duloxetine, the other is vortioxetine. Vortioxetine kind of came on the market later and is seen as the sort of newer generation compared to duloxetine. And I'll talk more about the data we have. So again, just a little bit more context on this applied area. There are effective depression treatments out there, but they are not effective for everyone. So only about, sort of 30 to 40% with major depressive disorder go into sort of a full remission after taking some of these medications. About 1/3 respond, but have some residual symptoms. And then another third or so sort of need additional therapy. The sort of first line treatment are some of the ones that you've probably heard about, at least in the US. They're known as Prozac or Zoloft, and these are called SSRIs. Duloxetine and vortioxetine are two sort of second line treatments. So for those people who don't respond to the SSRIs, duloxetine and vortioxetine are two of the medications that are then tried. They have adverse sort of side effects for both, nausea, et cetera. And both are seen to be effective compared to placebo. But very, very few trials have actually compared the two against each other, sort of head to head. And in particular, vortioxetine is also substantially more expensive, partly because it's newer on the market. And again, at least in the US medical context, which is complicated and expensive. So what we are going to be using today are, so Vivli is this data source that basically has partnerships with pharmaceutical companies to get their clinical trial data and release it for research use. So we have access to 4 randomized trials, each about 400 to 600 people. These were actually all 3-arm trials of duloxetine, vortioxetine, and placebo. We're focusing on that duloxetine, vortioxetine comparison. Some sort of fairly standard eligibility criteria. The MADRS score is sort of a common depression rating scale. This 22 to 26 sort of minimum is a fairly high level of depression symptoms. Again, sort of consistent with the role of these medications as kind of a second line therapy. The primary outcome in the trials, and again, this is one of the challenges in bringing in the EHR, which I'll come back to later, is going to be the change in that MADRS score from baseline to the end of follow-up. It's a little bit complicated with the change over time and the change, but a positive treatment effect estimate is going to imply that duloxetine is more effective than vortioxetine. So here's just a little bit of data from these 4 trials. So you can see some basic characteristics at baseline for the people in these. In general, the ages are pretty similar. Smoking status varies a little bit. One interesting, it's not super obvious in this table because I'm not showing BMI, but the weight distribution does differ somewhat, which could relate to efficacy because of body mass and sort of medication dosing. Interestingly, we looked into that a little bit more, and again, I'll say this as someone in the US, the higher BMI studies were done more in the United States and the others were more international. You can see the percent with anxiety differs quite a bit across, and the MADRS score and the HAM-A is sort of another depression rating scale. So anyway, this sort of gives you some basics. I'll come back to some of this as we go along. Okay, so let's formalize the statistics a little bit. So here, we're going to use A to denote treatment status. We have a set of covariates X, like those on the previous slide. Here it says continuous, although actually they don't all have to be continuous. And they aren't all actually. Y is going to be a continuous outcome. And so Y1 is the potential outcome under treatment. In this case, think of that as duloxetine. Y0, the potential outcome under control. Think of that as vortioxetine. So these 2 medications. S will be a study indicator. So in this case, we have K equals to 4 studies. And we're actually not going to really need to use it here because we have randomization, but potentially we could use a propensity score in each study to model treatment as a probability of the observed covariates. So the estimand at the bottom gets to sort of a little bit more formality of what we're trying to estimate here. So I referred to effect heterogeneity, modification, moderation. And this estimand at the bottom is, I didn't write it out in letters, but the conditional average treatment effect, it's often called the CATE, C-A-T-E, is this basically difference in potential outcomes given this set of covariates and here I'm conditioning on study. I'll come back to that later. So sort of for individuals in study S, what is this model for the treatment effect as a function of covariates? Here, this is sort of expectations and a difference for a continuous outcome. We could write this as an odds ratio or some other comparison between the potential outcomes. But again, so there's a few challenges here in estimating this. Number one, the biggest, of course, is that we don't observe Y0 and Y1 for each person. So each person is in one study And in that study gets either treatment or control. So for each person, we observe a piece of this. We observe one of these terms, sort of, and then we average across people. And the whole challenge here is going to be to sort of use the other people to help basically predict the missing potential outcomes. So this is kind of a quick summary of the, in some sense, fundamental assumptions. Some of the methods will also have sort of more modeling assumptions and kind of statistical models about observed data. Here, these are sort of more of the causal assumptions. So I don't know how familiar all of you are with this sort of thing, but I will just try to quickly summarize this. So SUTFA is basically saying that one person's treatment does not impact another person's outcomes, which is reasonable in this context. It also sort of assumes that there's sort of one, like if someone is assigned to take duloxetine in study S, that there is like one Y0 for them or Y1 for them, and that it's not sort of, it's like a well-defined value. The sort of truth is kind of a well-defined value. Unconfoundedness holds because we actually have randomization in these studies. Unconfoundedness is that there's no confounding, that the potential outcomes are independent of treatment given the covariates, which we know holds here. Consistency is sort of related to SUTFA. It's basically that we observe, if someone is assigned to treatment, we observe their outcome under treatment. If they're assigned to control, we observe their outcome under control. Positivity of treatment assignment also holds here, which is that in any given study, the people in that study have a positive probability of being treated or controlled. There's no one who in a given study who would never receive vortioxetine or duloxetine. Again, that's satisfied in part because like if there is someone who wouldn't be appropriate for one of the medications, they wouldn't be allowed into the trial. The sort of additional one, like those are all sort of standard for a single study. The assumption at the bottom is the one that sort of brings in this idea of how do we combine multiple studies. And sort of in the basic set of methods that I'll talk about, we basically have to assume that every person could have been in every study. This can be relaxed depending on the exact estimands and the exact methods, but we're sort of assuming that there is some overlap in the distributions of people across these studies. And we can, again, we could relax this, but for now, we're gonna keep it in place. All right, I don't see any hands or questions in the chat, but I'll pause for a second. Okay. So what I'm going to turn to now is to summarize, and some of you might be more experts in some of these things than I am. And so apologies for that. Again, I sort of think of myself as someone who brings statistical methods and helps think about their application in real world, mostly public health contexts. And so a lot of it is kind of using methods developed by others, which I really enjoy. But anyway, that's an aside. So I'm going to first talk a little bit about the kind of current thinking around some of these machine learning tools for estimating the CATE in a single study. And then we will talk about strategies for combining across studies, and then show some, not surprising for stats talk, simulation example. So again, in this context, we are particularly focused on some non-parametric machine learning sorts of approaches for estimating the CATE in a single study. And I'll talk at the end about sort of some of the pros and cons of that. There's actually, I think, some conceptual challenges with doing this, but I'll come back to that. The reason though the reason we are considering these machine learning methods at first is because of their flexibility. And so we sort of can use a number of covariates, and the methods will identify nonlinearities, interactions, et cetera, in a very automated way. As opposed to many of the parametric sorts of approaches, you have to say in advance, like, I think age is the moderator, and I'm going to put age in, in an interaction term or something. So this is, in some ways, I'm actually starting to think about this as almost more of an exploratory approach, where we can sort of use a number of covariates and use these methods to sort of identify where there might be some effect heterogeneity. there's two, so there's again sort of three types of methods broadly that are used in this context. Two of them basically separately model kind of the outcome under treatment and the outcome under control, and then combining those to estimate the Kate. The S learner does this sort of in a joint way though, where it sort of models Y as a function of the covariates and the treatment and interactions between those. The X learner estimates separate models of Y0 and Y1 and then combines them. So it's sort of a two models versus one model. I don't know why they are called S and X. So anyway. And then the third option is some work by Susan Athey and others that sort of directly tries to identify treatment effect heterogeneity. It's called causal forest, but it is sort of partitioning the covariates directly based on the effect heterogeneity itself. I'll sort of come back to some of these as we go along. We then again, in this context, are interested in combining across studies. And so we are going to consider a couple different approaches. One is what we'll call complete pooling, which you will not be surprised is not a good thing, but anyway, as a default, is to just kind of throw all the data together and pretend as if it's one single study. Again, not clearly, you can sort of probably tell that that's not going to work well, but it is done sometimes. The next approach is sort of well, we'll combine all the data, but let's at least include a trial indicator as a characteristic, sort of almost treat the trial as kind of a covariate in these approaches. The third approach, some of you might have heard of federated learning, and we're sort of using an approach based on federated learning. So federated learning is becoming more and more common, and it's used in contexts where data is being combined from a number of different research teams, but where the individual level data can't be all put into the same place. So the idea in federated learning is different teams sort of use models or sort of generate some output from their data, and then that gets combined in a common place, but the data itself doesn't get shared. So how that's been used in this context is that the Kate is modeled sort of within each study. So think of it like we have 4 research teams, one for each trial. They each fit a Kate model using one of those approaches I mentioned earlier. And then that models are sort of sent to the central place, and they're sort of sent across. So then each trial kind of predicts the Kates from each of those models. We would have 4 Kate models, and each study would predict the Kate using all 4 of those and then share those. So the sort of what would be shared would be like 4 times the number of people in each study to be then analyzed. And so then we have N by K Kate estimates, and those then get sort of modeled together to try to identify what is the variables kind of relating to treatment effects across all of the studies. That may or may not make sense. Oh, and I see the note from Carla. Oh, thank you for this makes so much more sense. Thank you for clarifying the S and T and X. And those are actually, so please read the chat. It's insightful. Okay, hopefully, and Carla, sounds like you must know about this world. So hopefully I'm not saying anything else wrong or anything wrong. Okay, we are going to compare these machine learning tools with kind of what I think would often be a default approach, which would be a sort of an individual patient data meta-analysis with random effects for studies. So sort of parametric outcome model, where you can see at the end of this equation, the X covariates by a treatment interaction, and that is allowed to vary across studies with that T sub S random effect. So we have some fixed parameters there, some random components, error, and in this case, the Kate model is going to be this parametric form of sort of intercept, like a main effect that has a random effect for study, and then an interaction, the theta, which is sort of the effect heterogeneity piece, and the T, which allows that to vary across studies. Okay. So again, sort of in this work so far, we are kind of trying to make sense of these methods that exist and how they might be useful for Kate estimation. So we started with a simulation. Again, this is really work led by my PhD student, Carly Lupton-Brantner. So here we are just setting up a scenario with 10 studies about the same size as the ones we have access to. And we're going to have sort of a variety of the sort of basically the main effects and also how much they vary across studies. And so that's what the sigma beta and sigma delta are showing, is sort of the variance of the main effect term, and then the sigma sub beta is the variance of the random effects for sort of the overall study effect, kind of the main effect of the treatment across studies. And then the sigma delta is showing the variance of the random effect for how much the effect heterogeneity. So if we have a moderator, kind of how much does the moderation effect vary across studies? If that variance is 0, it means that it's the same. So age has the exact same moderation across all studies. If it's 1, that is a more substantial moderation where the age treatment relationship differs, or the effect differs. Okay, so I'll just try to quickly summarize the takeaways here. So this is showing mean square error as a summary performance metric here. You can see we have the different colors relate to the different single study approaches. So the S-learner, X-learner, the causal forest, and then just the parametric meta-analysis. The x-axis sort of shows the aggregation approach. And then again, I said the y-axis is the mean square error. So you can see, not surprisingly, complete pooling where we ignore the fact that these are actually different studies performs quite poorly, not surprising. I'll go to the far right side of this figure next, which is the meta-analysis. And I want to point out that it looks reasonable, like relatively small mean square error. What's kind of hidden there is that, not surprising, what's happening underneath that is that if the meta-analysis is specified correctly, like if the model is true, then the mean square error is very small. If the model is wrong, the mean square error is very high. So that's not shown in this figure. So, but sort of there's a lot of variation across the settings. In terms of looking at the colors, you can see that the causal forest tends to give us the smallest mean square error, maybe because it is sort of targeting that causal effect directly rather than sort of going through the other models. Not huge differences though. And then in terms of the sort of x-axis, the ensemble forest, where we are kind of, that's this sort of federated learning approach where we're doing that. We're sort of fitting the model in different studies and then combining them and then getting the predictors again, seems to work the best. The trial indicator, though, also does very well and is very easy. Like the trial indicator approach is like pool all your data. So if you can pool your data, pool all your data and at least account for study. And that gets pretty good performance, all things considered. Again, so some other takeaways that weren't all shown exactly in that figure. It's always hard to show full results in a 45-minute talk. But not surprisingly, underneath those dots I showed, as the variability of study coefficients increases, so as there's more heterogeneity across studies, the mean square error increases. Again, not surprising. The ensemble lasso, I didn't really mention lasso, but we used an ensemble lasso approach, performs really well for like true linear Kate models, but not as well when they're nonlinear. Sort of similar for the S-learner approach. And again, sort of the causal forest was the most consistently well-performing, and then sort of the pooling with trial indicator or the ensemble causal forest approach. So we took those lessons to apply them to for randomized trial data. So in particular, I'm going to show results from using the causal forest and then pooling with the trial indicator. So here, and again, I'm going to have a slide and a few slides that is sort of all the caveats of all of this. So stay tuned for that. But so this shows across the, 2,000 people, 1,600, 2,000 people in this combined data set, the individual Kate estimate. So again, we don't really believe that these are individual treatment effects, to be clear. It's not sort of like this is the effect for Joe, but think of it more as sort of the effect for people with these different underlying covariate distributions. And I want to highlight that you can see here first that the average is right around 0, not a lot of effect. And these intervals are very, very wide. So even in this case where we're combining 4 trials that are each, average size for this world, our Kate estimates are highly variable in terms of, and so again, this sort of showing like we don't actually have a lot of information to do this. What we can also do though, this is sort of not super satisfying because it's like, okay, but how do we make sense of this? And so sometimes what is done is then to use sort of what's called an interpretation tree for the causal forest, where you kind of fit sort of a cart type tree model to the causal forest Kate estimates to sort of identify like what are the variables that are kind of popping up as most important for determining effect heterogeneity. And so here you can see that age less than or greater than 30 is sort of the first split. And then below that sort of weight pops up, this base MADRS score pops up, and those are sort of the main variables, sort of age, weight, and the MADRS score. But, again, sort of the original goal here is like, well, we'd like to ideally, be able to use this to help inform treatment decisions for people. This is also sort of hard to think about, like, okay, are we really gonna, sort of follow this? So again, I'm sort of moving towards thinking about partly as mostly just exploratory. So, so then we might think, okay, let's dive a little bit more into age. And so we can then use these Kate estimates to look at the actual Kate estimates across different age ranges. You can see the colors just show which study the people are from. You'll notice immediately the red lines. So this is like the messiness in combining data. This was not in any of the documentation, but once we actually got the data, we realized that everyone in that study was, in these different, was like, 24 or like 25, 35, et cetera. And it's just that they coded them sort of giving everyone within a band that value. So again, the real data is messy in ways sometimes that you don't expect. So don't think of those red lines. Not everyone really was 25. They actually had spread around that. But you can see here, sort of, this is where you can say, okay, like, do we see, maybe if I squint, there's a little bit of indication of some effect heterogeneity across age, but not really. And remember those super wide intervals I showed 2 slides ago, each of these dots is actually measured with substantial error. So, kind of unclear what to make of that. We could, though, also look, say, and again, sort of these are the things we can do with the data. I'm not saying we want to do it, but we can, and people are doing, this interest in effect heterogeneity is sort of all over the place. So we could look at these Kate estimates across age and BMI deciles, and we could sort of see, okay, do we see larger Kate estimates? There's that one upper left yellow kind of biggest estimate, but again, lots of underlying variance that's not really being shown here. So, okay, let me, great. Oh, I have plenty of time. So, but I want to take a few minutes to kind of like reflect, because honestly, this is partly my like process in doing this work. I think there's this, again, real interest in a lot of fields for like, oh, we really want to identify all of these interactions and sort of all the ways, like, estimate these Kate models really well. But I think translating those into practice is still really complicated. Like, we can have our big fancy statistical model that sort of gives us these Kate estimates, but how do we summarize them in a way that is responsible and kind of gives information that's helpful? And sort of one of the things I'm struggling with, curious if any of you have thought, if you work in this area, is that, we can use these Kate models and we kind of come up with individual estimates, and then we can sort of look at different factors that relate to that. But in reality, then, if we're really going to be sort of like looking at sort of, well, like, does age matter? Does BMI matter? Does, et cetera? Maybe we should just have started with age, or maybe we use the Kate approach to sort of identify which variables to focus on, and then, go back to like a fairly basic interaction model with age. And so again, that's sort of partly where I've been thinking about exploratory and confirmatory analyses. Again, there's also huge uncertainty in these Kate estimates. Also, I haven't touched on this, but some of Carly's current work is that, in reality, the goal is to sort of be able to use this to help make treatment decisions for sort of future people. But of course, future people are not in a study. And if we have variation in these models across studies, how can we make conclusions about that? Sort of in some ways, like really what will be, it's like we want to figure out what is the sort of cross study core effect heterogeneity and separate out the study specific pieces so that we can sort of understand maybe more what's like biological and will carry through into the future versus what kind of just is study specific. And then this last bullet is, I will say from working on this in a number of areas, like I don't know of very many examples where there is, it turns out there sort of seems to be true effect heterogeneity. So I think that's also a challenge, is we also, again, just want to be really careful that we're not sort of overstating effect heterogeneity that doesn't really exist. There are some examples clearly where there is true effect heterogeneity, but at least in mental health, really it seems like at least the variables that we have to predict the heterogeneity don't seem to be very useful at doing so. And that's true not just in this data that I'm showing you, but across a whole number of applications in mental health. And I didn't even really talk about the electronic health record data, so I pulled you in with that title. But I'm happy to talk more about it. I think fundamentally what I have been thinking a lot about is one of the challenges is, again, it's sort of one of these like easier said than done, because we have access to electronic health record data at Duke and at Hopkins, but they're messy. Even, thinking about, and anyone who works with this sort of data knows this, even just thinking about how do we define a cohort of individuals? People come in and out of a particular healthcare system. So someone might be at Hopkins for a month and then we never see them again. How do we sort of think about the biases that come in then when we are sort of looking at people within that sample? The other major problem is that in the US at least, most of our electronic health record systems don't collect any sort of like symptom data. And so, the randomized trials collect outcomes that are things like symptoms. The electronic health record data, we don't have that. We tend to have service utilization. We might have emergency department visits. So Duke, for example, is starting to collect this one symptom scale called the PHQ, but it's literally 2 items, the PHQ-2. And so we basically are now also working where we're realizing that we can't just start combining the data yet, because our outcomes are just completely different outcomes. But a colleague, Chang Nguyen, is starting to work on some methods to try to have models that sort of allow for different outcomes and have some parameters that are shared across outcomes or shared across, and then some that are outcome specific. So all of that is to say that the EHR data, like we can have, like I could write out the methods to, like if we had the same outcomes, the methods extensions are not that hard. The problem is that the data is incredibly messy, and we have these complications about different measurement and things, which make it that the methods we might have sort of laid out before digging into the data, it turns out are just not going to be useful because of the data complications. So sort of overall, I would say, if you are in a scenario where you have multiple trials and you want to investigate effect heterogeneity, the sort of pooling with trial indicator and ensemble forest tended to work well. Not surprisingly, parametric approaches struggled when the true Kates are nonlinear. And I think one other thing sort of I think the field needs is more diagnostic, sort of how do we help people sort of think through which methods make the most sense in their data set. But again, lots of limitations, and this is all very much a work in progress. So certainly more scenarios we could examine. Again, there's this sort of tension about trials. And I will say, actually, when we've been doing reviews of the literature on methods for combining data from multiple studies, this point about whether the estimand is study specific or not is actually often quite buried. And I think this is something we need to think a little bit about conceptually, is sort of what are we trying to estimate? Do we want a condition on study, or do we want to be able to have some sort of average across studies, but then what does that mean? And again, that is in papers that often kind of gets buried. So lots more work to do. I just want to highlight here, these are some of the key references. So the Athey paper at the top is the causal forest kind of original paper. The second one is a nice review paper that Carly wrote for a special issue of Statistical Science that is, again, sort of overview of the methods out there for integrating these sorts of data sets. And then, yeah, the Kunzel talks more about the S-learner and X-learner sorts of things. And then the TAN is more of that federated learning. And I just realized that I thought I had added another. We also now, while we have the Q&A, I'll put it into the chat. We have a paper on arXiv and under review that is sort of the paper version of like the simulations and the application that I presented today. So I can also share that.